Okay, thank you. Hi, everyone. I'm Xiaomin, just defended my PhD dissertation last November, and Lance is my supervisor. Today, I'm delighted to have a brief introduction of the PhD project in 10 minutes. Very ambitious, right? As you can see from last presentation, he gave a big picture for the research scope on food exposure or intake biomarkers. And what I have done is one part of it. So I'm going to talk about how we applied untargeted metabolomics to discover intake biomarkers for heat-treated foods. Last year, we were invited to an interview with social media, and our research output was posted in TV2 Lorry. I use this picture here for you to better understand the project. We aimed to develop a method to review what people have eaten and why we'd like to develop this new method to assess dietary intake. The challenge in nutrition research is that there is a high degree of uncertainty when it comes to the accuracy of participants reporting back what they have eaten. And later, the biomarkers of food intake, BFIs, come out as a promising solution because they could offer greater objectivity and reliability. The BFIs are food-derived metabolites that they are present in biological samples. Furthermore, the PhD project is one part of a big project, football, as mentioned by Lars. We have a common aim to identify existing and novel biomarkers of intake of highly consumed food. I was dedicated to tubers. Use the laser point. I was dedicated to the tubers, including potatoes. On the other hand, the skimmed milk powder was selected as another type heat processed food. So the corresponding experimental work at two major paths consists of the PhD framework. We can call potato study for part 2, milk study for part 3, and part 1 is the systematic literature search to identify existing markers in the published paper. I'm not going into details for this one because of limited time. So this slide, I provide an overview of metabolomics workflow from the study design to biological sample to biological interpretation. Next, let's dive in the study design. Potato study is an RCT design. 4 test meals were provided to participants, boiled rice as the control group, and 3 different potato products as the intervention groups. For the sampling, the baseline and 7 postprandial urine samples were collected. Regarding the milk study, is a design of the rat study. All 5 dyes in 5 groups contained 20% skim milk powder, which consisted of different heat and or hydrolysis treated fractions. So see this table. After feeding the test meals, 24-hour urine samples were collected after 1 week. So there are 2 factors, heat treatment and hydrolysis in terms of the diet. We only focus on the heat treatment. We use the LC cutoff mass platform for urine profiling. Regarding the data preprocessing, MZ mine was conducted to extract the important features. In terms of the data statistics, both ANOVA and PSDA were used for classification and variable selection in potato study. For the milk study, we applied the ASCA for data decomposition, and the dataset after subtraction of hydrolyzed was applied for PSDA analysis. After this procedure, a list of discriminating metabolites was selected to reflect intake of heated food. And then we go to the next step, metabolite identification. As we know, metabolite identification is the most challenging and time-consuming part. So I'd like to briefly share the strategies of identification applied in this project. For the known compounds, they can be identified by searching against a database, or including our in-house database and the spectral libraries. However, most metabolites are unknown compounds, so we use the QTOF and the LTQ or B-trap to obtain mass pattern for data annotation, and then we run the standards, analogies, or the synthesized metabolites conducted by ourselves to reach the identification level 1, or we can at least confirm the class or co-structure of the unknown compounds. So from my experience, we always need to explore the information on chemistry and biochemistry by applying the tools or looking at the published papers to facilitate identification. So that's all I have to say about the method for 2 studies. I know they are present in a little fast way, but you could look at our published papers for details if you are interested. Now, turning to the exciting moment, many focus on the characterized markers. So let's start to answer our research question. Could we find putative BFIs for heated potato products and markers for heated milk powder? Yeah, the answer is yes. 14 metabolites were tentatively identified for deep fried potatoes and 19 metabolites for the heated milk powder. As they are formed by heating, they are not specific metabolites for heated milk, for heated potato and milk powder, but the particular pattern together with the more specific compounds derived from the food itself could consider to be as BFIs for heated food. We also found 3 common markers in 2 studies which support the robustness as candidate BFIs for heated food. Furthermore, the tentatively identified metabolites, especially the AGEs, advanced glycation end products, have not been reported in urine. You may be curious about the AGEs. I will talk it later on. We also propose their formation during the cooking processing and biotransformation after consumption or feeding, which I'm not going to describe here, but they are documented in the papers. So the work we have done is focused on the discovery of putative BFIs. So definitely more studies are required on BFIs validation. For example, the performance on dose response, robustness, reliability, or the reproducibility, according to the validation scheme outlined by Lars and other co-authors. Another interesting perspective is physiological consequences of dietary AGEs. So AGEs, advanced glycation end products, are formed by the reaction between sugar, for instance, dicarbonyls, and amino group, for example, protein, to form glycated compounds. This figure shows the impacts of AGEs on human health, for instance, the impact on inflammatory factors and glucose metabolism related to development of diabetes and diabetic complications. And the formation of AGEs can be formed in our body, and they can be consumed from food. As I mentioned before, we found 12 new AGEs which have not been reported before. Furthermore, the new AGEs are far more abundant than the common and representative AGEs such as CML and MGH1. So this result indicates that the new AGEs may have potential higher biological reference. Therefore, we would like to go a little further to investigate whether the new AGS excreted in urine indicate the risk of development of diabetic complications, and the level of AGS in urine could be modulated by dietary habits. So there are our major perspectives based on the result from this project. Acknowledgement. So I'd like to thank my supervisor Lars for the guidance on my scientific development, my colleague Christian and collaborator Marinka and Jie for their professional assistance on instrument data annotation. Special thanks to Catalina for her contribution on the co-design and conduction of the human study, Melina for the animal study. We acknowledge the financial support from football and Chinese scholarship on this project. Also, a big thanks to my wonderful colleagues like Yan, Henrik, Mu Yao, Simon, and so on. Thank you